Welcome to this lecture on immunology. Today, we are going to explore one of the most fascinating and crucial components of our immune system, the antibody. Imagine your body is a highly secure fortress, constantly under threat from invisible invaders like bacteria and viruses. How does your body identify and neutralize these threats before they cause catastrophic damage? The answer lies in a microscopic, custom-built defense mechanism. In this session, we will uncover the elegant structure of antibodies, understand how they function as biological guided missiles, and learn the mathematical principles behind their size and binding capabilities. Let us dive in and discover how these tiny proteins keep us alive every single day. To begin our journey, let us understand why antibodies are so vital. When bacteria or viruses breach your body's outer defenses, your immune system needs a way to track them down. It does this by producing millions of tiny, Y-shaped guided missiles called antibodies. These are not generic weapons. They are incredibly smart and custom-built to recognize and lock onto one specific type of intruder, much like a unique key fitting perfectly into a specific lock. Once they attach to the invader, they act like giant neon signs, shouting at the rest of your immune system's heavy artillery to come and destroy the threat. Without these brilliant little defenders, our bodies would be completely blind to the specific pathogens that manage to sneak inside. Now that we understand what antibodies do, let us look at how they are built. In formal immunological terms, an antibody is also known as an immunoglobulin. These are large, Y-shaped glycoproteins produced by specialized white blood cells called plasma cells. If we zoom in on the basic structure of an antibody, we see that it consists of exactly 4 polypeptide chains. There are 2 identical heavy chains, which form the main structural core, and 2 identical light chains, which sit on the outside of the Y's arms. These 4 chains do not just float together, they are firmly held in place by strong covalent disulfide bonds and various non-covalent interactions, ensuring the antibody remains stable as it travels through your bloodstream. Building on that structural foundation, let us examine the specific regions of these chains. Each of the heavy and light chains is divided into 2 distinct parts, a variable region and a constant region. The variable regions are located at the very tips of the Y shape. As the name suggests, these regions are highly diverse, which allows the immune system to recognize billions of different antigens, the foreign molecules that trigger an immune response. This is where the actual binding to the pathogen occurs. On the other hand, the constant region forms the stem of the Y. This part does not bind to the antigen. Instead, it determines the biological class of the antibody and interacts with other immune cells to execute the destruction of the neutralized pathogen. Before we move on to the mathematics of antibodies, let us see this structure with a concrete visual diagram. What you are looking at is a classic Y-shaped antibody monomer. Notice the 2 longer heavy chains forming the inner core, colored in blue, and the 2 shorter light chains on the outer edges of the upper arms, colored in orange. See those small black lines connecting the chains? Those represent the crucial disulfide bonds that staple the entire protein together. At the very top, the tips of the arms are labeled as the variable region, which is the antigen binding site where the antibody locks onto a virus. The rest of the structure, the stem and lower arms, is the constant region, which signals the rest of the immune system to attack. Having visualized the structure, we must ask, why is understanding this so critical? The structure and function of antibodies form the absolute foundation of how we survive in a world filled with deadly pathogens. In real life, this concept is the exact reason why vaccines work. A vaccine safely trains your body to produce these specific antibodies against diseases like COVID-19 or measles long before you ever get sick. Furthermore, scientists use this deep structural knowledge to engineer monoclonal antibodies in laboratories. These engineered molecules are used as life-saving treatments for cancers, autoimmune diseases, and severe infections. Without antibodies, even a simple paper cut could introduce bacteria that would multiply unchecked, leading to fatal infections. They are our ultimate targeted defense mechanism. Now that we understand the biological importance, let us look at the mathematical side of antibody structure. Because a basic antibody is a symmetrical Y shape made of two identical heavy chains and two identical light chains, we can easily calculate its total mass. The formula is M total equals 2 times mH plus 2 times ml. Conceptually, this just means you multiply the weight of each chain type by 2 and add them together. In this equation, M total represents the total molecular weight of the basic antibody monomer, usually measured in kilodaltons. mH is the weight of one single heavy chain, and ml is the weight of one single light chain. If the weight of the heavy chain increases, the overall size of the antibody increases, which directly affects how easily it can move through body tissues and blood vessels. Let us apply this formula to a real-world scenario with an example. An immunologist is studying an IgG antibody, which is a standard monomeric immunoglobulin consisting of a single Y-shaped unit. We are given that each heavy chain has a molecular weight of 50 kilodaltons, and each light chain weighs 25 kilodaltons. We need to find the total molecular weight. First, we recall our formula and substitute the given values. mH becomes 50 and ml becomes 25. We multiply each mass by 2 because the symmetrical structure requires a pair of each chain type. 2 times 50 gives us 100, and 2 times 25 gives us 50. Adding these together, we get a final total molecular weight of 150 kilodaltons. This relatively large size means the IgG molecule remains primarily in the blood and extracellular fluids to hunt down pathogens. This brings us to another crucial mathematical concept in immunology: valency. Valency is simply the total number of antigen binding sites available on the entire antibody complex. The formula to find this is V equals 2 times N. Conceptually, this makes perfect sense because every single Y-shaped monomer has exactly 2 binding sites at the tips of its arms. Therefore, the total valency is just 2 times the number of units. In this formula, V represents the valency, and N represents the number of basic Y-shaped monomer units joined together in the final structure. If the number of units increases, for example, when antibodies link together to form polymers, the valency increases. A higher valency allows the antibody to grab onto more pathogens at the exact same time, making it highly effective at clearing infections. Let us see this valency formula in action with a different class of antibody. During a bacterial infection, the body produces IgM antibodies. IgM is unique because it is secreted as a pentamer, meaning it is made of 5 basic Y-shaped monomer units linked together. We are asked to find the total valency of this complete IgM pentamer. We start by identifying our variable N, which is 5. We substitute this into our valency formula, giving us V equals 2 times 5. The calculation reveals a final answer of 10 separate binding sites. Physically, this incredibly high valency makes IgM exceptionally effective at grabbing and clumping multiple bacteria together at once, a vital process known as agglutination that helps clear infections rapidly. Now that we have calculated the valency of the IgM pentamer, let us prove just how massive this polymeric antibody truly is by deriving its total molecular weight. This requires a multi-step approach. Our first goal in this chunk of the derivation is to find the weight of just one single Y-shaped unit of the IgM antibody. We are given that the heavy chain mass is 65 kilodaltons, which is slightly larger than the IgG molecule we saw earlier, and the light chain mass is 25 kilodaltons. We substitute these into our standard monomer equation. 2 times 65 gives 130, and 2 times 25 gives 50. Adding these together, we establish that a single IgM monomer unit has a total weight of 180 kilodaltons. We will use this foundational result in the next step. Continuing our derivation, we will now calculate the final mass of the complete IgM pentamer. We take the single monomer weight of 180 kilodaltons that we just found and multiply it by 5 to account for all the units present in the star-like pentamer structure. This gives us 900 kilodaltons. But we are not done yet. These 5 units are stapled together into a cohesive ring by a special connecting protein called a J-chain, which weighs 15 kilodaltons. We must add this J-chain mass to our total. 900 plus 15 gives us a final calculated molecular weight of 915 kilodaltons. This mathematical proof demonstrates that IgM is a giant macroglobulin. Because of this massive size, it is strictly restricted to the bloodstream, where it acts as a phenomenal first line of defense. We have covered a lot of ground today, so let us summarize the key takeaways from our lecture. First, we learned that antibodies are highly specific, Y-shaped guided missiles meticulously built from heavy and light polypeptide chains. Second, we saw how their unique structure features variable regions that act as ultimate biological sensors to bind specific invaders, while their constant regions act as an alarm system for the rest of the immune system. Finally, by calculating their valency and molecular weight, we proved how different classes of antibodies, like the nimble IgG or the massive IgM, are perfectly engineered to protect our bodies in diverse ways. You now have a solid understanding of antibody structure and function. Excellent work today.